Okay, so the idea of today, I think many of you have already heard at least some of the gist of what this presentation is going to be about. But the idea of the talk today was basically just to introduce you to the project that Morten and I wrote last summer, and then got funded sometime in December, called FindingPheno. And Morten is the one who's going to claim should deserves all the credit for the fantastic name. So the Finding Pheno is basically a response to a EU research and innovation action call, which asked for new methods and novel methods to be applied to discover genotype-phenotype associations from multi-omics data. And was quite an open-ended call, and our interpretation of it was, given all of the data that we have from Whole Food and from other similar projects that are starting to sprout all over the world, there was quite a niche for developing computational tools that would eventually could be used for integrating microbiome data and other microbiome related genomic observations from it. And so obviously it's a large consortium. It consists of 8 different partners, including the University of Copenhagen. And this is a little one step behind the innovation action call. So we do not actually need to demonstrate uh, actual application in the industry, but we do need to show that the industry would benefit from it. And therefore, there are also the industrial partners here, Kyogen and Christian Hansen and North Bio, whose logo I couldn't find. So what do classic genomic studies look like? Classic genomic studies, and there is no dearth of genomic data across all of the last 15, 20 years of the explosion of sequencing. There is absolutely no dearth of genomic data. But the classic genomic studies looks at a single biological process, may be variation in the genome or variation in expression, protein, epigenome, metabolome, or what you may have, or sometimes also looking at differences or changes in the microbiome. So probably the metagenome and the metatranscriptome or the metabolome, and looking to see how any of these affect a phenotype that you might be interested in. So let's say you're interested in the longevity of a particular individual. You want to collect all of these kinds of data, and then you want to look at how each of these particular genomic processes could be related to how long a person lives. And traditionally, this is done by putting each of these different data sets in a silo of their own. And recently, that's not true anymore, but traditionally, this is definitely the case where each of these data sets are looked independently. So you want to see how these data sets correlate with changes in whatever phenotype you might be interested in. And then you're going to find some parts of these genotypes. So maybe some gene that's expressed more or less in a particular individual, depending on their phenotype, or some SNP that might be present or absent. And you make a catalog of all of these. And in many, many cases, you want to look to see how these different categories are associated with it. So that's the classical association study. Does this SNP appear more often in people who will be longer or shorter? Or does this gene get expressed higher or lower? But that's ignoring the fact that almost all of our biological knowledge indicates that none of biology is as straightforward as it looks. There's a lot of redundancy, feed forward, and feedback mechanisms between these biological processes, which are basically being ignored for the ease of modeling. And so we are going to talk about some of the shortcomings of these multi-omics methods. There are going to be 3 major issues that we are going to focus on. The first issue that we are going to talk about is aphorism you've heard probably hundreds of thousands of times if you've done any kind of genetic studies or any kind of model, statistical modeling, is correlation is not causation. And what do I mean by this? Let me demonstrate it using some examples. So this slide's going to take, it took me a couple of minutes to load this slide when I was showing this, but apparently my computer's gotten faster. Um, so this is the latest information from EBI on across the human genome, the various loci that have been implicated in different diseases and related traits. Some of these are related to height as well. So not just diseases and anthropomorphic traits, right? Metric traits. And so here you can see there's so many traits, so many locations across the genome, each of them having various different effects on it. And I was a little bit surprised. I counted across this and I said, yeah, I can see the 22 autosomes and the X chromosome. I thought they had ignored the Y, but obviously the Y is a rubbish chromosome. You can see it at the end. It has no associations at all. Nobody studies the Y. Nobody cares about it. But of all of these findings that you've had across the entire genome, how many of them really get translated to actionable intelligence, which we can use to maybe treat a disease or maybe use it to look at the biological pathways that are indicating how much do we better know function when it comes to biology. And this is another example of a similar kind of research. This is only up till 2016. This is the number of SNPs that have been associated with various traits here. And you can see that the number of SNPs are massively exploding, but all of these have to go through a particular process before we can get through the drug compounds. And unfortunately, when we get to the drug discovery or drug uh, or any kind of molecule that we care about, we need to get additional information. In panel C, you can see that all kinds of additional information is now taken into account to make sure that the drug target that's being developed does not actually uh, cause any side effects, at the same time also related to the trait that you care about. Why don't we do this at an earlier stage? And that's where our we want to get better models that will actually help us integrate this data at an earlier stage. So this is the dream scenario, right? You have a rain and you that leads to wet floor. Super easy causation, right? And but unfortunately, as with biology and many other complicated systems, this is never going to happen. This is like a statistician's dream, but that's never. This is never the case in real life. And this is most of the problems that we encounter are due to a problem of scale. This is a schematic of the human microbiome project, and you can see that here they have samples from across a lot of tissues and a lot of places in the body. And here they're collecting different kinds of data as well, as you can see from HMP2's panel. And there is also some temporal aspect to the data. When you put all of these together, the amount of data that you need to interact with each other and the interactions that you need to measure is a combinatorial nightmare. So real data looks more like this, right? This is not biology. This is someone trying to figure out how to make Afghanistan into a land that can be governed properly. Clearly, this problem exists outside biology as well, but there is a lot of arrows here which talk to each other. And there is a lot of interactions that need to be taken care of. And this is true, obviously, in biology as well. And so there are a few solutions that we have been thinking about and that we want to apply to some of the data that is collected through other projects. And some of them include structural causal models, which basically let you go beyond correlation and assign a direction to the arrow, saying this SNP is the reason why you see a higher or a lower age in some of these populations. Then there is also Bayesian hierarchical models where we're going to integrate information from different layers at different stages and somehow use this information in a sort of a repetitive manner to bolster our understanding or our belief in what these genotypes lead to in terms of the phenotypes. And finally, there are also statistical learning methods, including some conditional variation autoencoders and stuff like that. I'm not going to go into technical details of any of this. If you're interested in any of them, please do feel free to come and talk to me. I know at least about one of them. I don't know about all of them. So obviously, this being a consortium-based project, there are lots of partners. This work is going to be done here at KU and at the Champalimau Foundation in Portugal, and through Gonzalo Polavieja, who's a PI there. So, what is the second shortcoming of multiomics methods? Second shortcoming of multiomics methods is less of a shortcoming, more of a convenience that we have been using all this while. Is that we use a steady state assumption. So we assume that the data we collect is what the system behaves like usually, and this is almost certainly not true. Uh, Pardon me. Under the steady state assumption, we assume that a system, no matter where it starts, and the moment we are collecting a particular piece of information, like the genomics data, we are collecting at the state where the system is entirely in an equilibrium state. So what we're collecting is reflective of what the system will look like in the long run. And this might be true, but we know that biology also, many other systems don't behave like this. There is quite a bit of action which goes on before a system reaches steady state, and different parts of the system may not behave equally well. Does this mean all of the data that we've been collecting and working on is useless? Certainly not. The data that we've been collecting and using is useful because we are looking at a population of cells. So, on average, most of these cells are going to be in the steady state because the dynamic state doesn't exist for that long. And when I say steady state, I mean both in terms of time, like development stages of an organism, or in terms of uh, what kind of season we are looking at, or in terms of whether the transcription of a particular gene has already reached this steady state. But in order to accomplish some of these temporal variations that we might see in the microbiome and the host, we which, like I said before, might be across developmental stages of a chicken, because I know we have data from that, or across seasonal changes, like they do in the Human Microbiome Project, uh, is going to be focus of one of our collaborators, Leo Lati in the University of Turku, using some models called the Ornstein-Uhlenbeck models. These are basically simple Brownian motion models, but they have some level of correlation between what changes might happen over time. And in terms of spatial variations, this is going to be the main focus of Istvan Schering at the Center for Ecological Research in Budapest. And he's going to use two different approaches to it. One of them is based on predator-prey models, also known as the Lotka-Volterra models, which basically looks at the interaction of predator and prey and trying to incorporate this kind of an idea into how the microbiome behaves over space, is something that we are also interested in. And the other idea that is going to be used is based on game theory models on something called shared community goods, where you can think of host and the host metabolites as as community products which are used by all of the microbiome. And then you're going to think about also the products which are generated by the microbiome as something that's a resource that's shared across all of the community. And clearly there are some advantages to producing these shared goods or using them, and there are different evolutionary strategies about how different microbes can come together, or whether it's better to take advantage of this, or whether it's better to contribute, or what kind of proportions of contributors versus users is required for a stable system. And much of this work, at least some of this work, is based on this person's picture here. I'm not sure how many of you can recognize who this is. I'm going to give you a hint in a minute. Yes, it is John Nash. It's John Nash's equilibrium theory kind of points at some of this as well. So that's going to be used in some of these. Finally, the third shortcoming that we are going to talk about is there have been so many studies on many of these processes, that biological processes that we are interested in. For example, just recently we submitted a paper on on behavioral aspects and physiological measurements in a particular population of mice. And I know for a fact there have been other labs that have been working on precisely the same mouse and very similar data in terms of phenotypes that they're interested in. But at the same time, we did not really talk to each other or use the previous results to kind of uh, hone into what the real meat of the problem is, where the genetics might play a role in some of these phenotypes. And this is widespread in biology. Every person who generates a data set seems to come in thinking that their data set is the most unique and the best data set ever produced, which is possibly true. But at the same time, there is no reason to discard all of the previous information that we have gathered over time. So over the last 4 or 5 years, there has been a concerted effort, at least in human health, to better leverage some of these previous findings and trying to improve our understanding of genotype-phenotype associations in light of these findings. So we are going to actually use some of these methods as well, and we are going to use them on two different problems. We are going to look to see if previous studies of the same systems in terms of both phenotypes and the model systems we are looking at can be used to identify or prioritize different pathways or different genes that might be of more interest than other parts of the genome, using some statistical methods such as Bayesian variable selection, or adjusting the priors based on what kind of or modeling adjusting the priors based on a model for what kind of different parts of the genome might be useful or not. And this work is going to be primarily focused here in Copenhagen with Fernando and me. Uh, and finally, the part of it which is quite exciting is we can talk all we want about developing different models, clearly validating these models and applying them to real systems, and checking that these work and figuring out what kind of improvements we can make based on any advancements we make, is going to be quite crucial. And here is where uh, we are going to work with Christian Hansen and Rob Finn at EBI, and clearly their knowledge on and involvement in Holo Food, where many of these data sets are being generated, is pivotal. And then we can basically use these data sets to train our models, make them better, and kind of figure out if we can basically pull more information out of these data sets than what would be done if we're using simple pairwise association models or any kind of a posteriori combination of the information. And so this is going to be applied to many different data sets. Chicken, salmon, you already know from Holo Food. If you don't know from Holo Food, now I'm certain all of you know this from Holo Food. You're in CH meeting. And there is also maize from Back for Crop, which may or may not end up being used in the project due to some issues. But there is also public data that's being generated, and much more of it now. There is a lot of data on tomato, honeybees, wheat, maize, fruit fly, you name it. Microbiomes are now basically taking over, have become an integral part of many of the multi-omics genomic studies. So there is no real dearth of data sets. And what do we hope to achieve by the end of it? Instead of looking at the previous picture that I showed you, where we are looking at classic association studies, the hope here is that uh, clearly I've simplified this model far too much because I've ignored all of the arrows that could be drawn here, so as to not make it like look like this picture similar to fixing Afghanistan picture. But the hope is that we would be able to, in some sense, integrate the data from the host and microbiome together in an analysis framework that's aware of these variations rather than combining them a posteriori, including both evolutionary analyses and time series analyses, which allow us to model some of the spatial and temporal variation that we see in the data set. And hopefully, when we do this, we can get better insights into what kind of genotype phenotype associations exist, what kind of redundancies might exist in the data, in the biological pathways, and thus hopefully leading to better intervention strategies. Not drugs, but hopefully, in this case, some way to modify the organisms that we are studying, chicken and salmon. And obviously, the ulterior motive, which nobody knows, is we want to make a lot of money. But we'll see. Never going to happen, is it? With that, I'll take any questions and thanks for listening. But before I take questions, we have a project and outreach manager position open for the project, for the Finding Pheno project. It starts fairly soon, sometime in the middle of next month. So please do get in touch with your networks, or if you have people that who might be interested, might be interested in such a position, please ask them to get in touch with me or Morten.